COVID-19 triggers a cellular process that contributes to brain aging. And the good news is that now we understand this process and we can reverse this. So what we have found is that SARS-CoV-2 can infect the brain and induce a process called cellular senescence that is contributing to the aging process. We now have therapies to target specifically senescent cells. And these compounds we've found that can eliminate selectively the senescent cells that are induced by the virus in the brain. One disadvantage of research in the brain is that it's difficult to do clinical trials, especially when it comes to viral infections. So human brain organoids are a key tool to address this gap.
An organoid is in essence a small mini version of an organ in the sense that it has both the architecture, the cellular makeup, and most importantly the function of the organoid in question. And that allows us to do two important things. It allows us to study the interaction between the genetic background of an individual and disease. And because we can make them at scale, we can use them for drug screening and therapeutics identification and testing. What Julio has done is quite an amazing feat, really, to use human brain organoids and other different models to show that this virus induces a process that may accelerate aging. I think where he really stands out is to show that you can use them for screening drugs. And I think going forward, that's not just going to help people that have been infected by viruses that infect the brain, but could also be useful for studying the role of senescence in Alzheimer's disease, neurodegenerative diseases, and maybe a whole host of other conditions where senescence is a driver.